Okay. So hello everybody. I'll just share my screen and we can begin with today's lecture. And if your fellow batchmates they join in between, so we'll just let them in along the class. Okay. So let me begin with this week's lecture class actually. Just a moment. So I have not edited that this is for week 8. I hope I have done that editing in this particular one. Yeah. Okay. So hello guys, if you have been attending my classes or these practice sessions that I have been taking for this course cell biology, you might be aware of how we are conducting these classes, right? For people who are new, I will briefly introduce myself. I am Arveen Kaur. I'm a PhD scholar at the Department of Biological Sciences and Bioengineering at IIT Kanpur. Also, along with this, I am a teaching assistant for this course that is cell biology, cellular organization, division, and processes that is currently being taken by Professor Shikha Laloria from IISc. Okay. So, moving ahead with this week's lecture, so it is going to be the final practice session pertaining to a particular week, right? Along with this, I want to let you guys know. Just a moment. Let me just share my screen again, okay? Because I am not really getting that window here. Yeah, so I'll share my screen again. Okay. So, as I was saying, are you trying to say something? Okay, if that was intentional, could you please repeat? Anyway, we'll let that go by. So this will be our final practice session pertaining to a particular week's lectures, right? So pertaining to week 8 lectures, we'll be discussing the previous year's assignment that was given for this particular week, right? Along with that, we will also discuss some concepts that have been discussed in this particular week. In the second part of this particular session, what I'll be doing is that I have basically prepared from reference from a particular review paper. I have prepared a lecture on the various kinds of cell deaths that are possible, right? So that is what I told you in the previous session. I'll be discussing this week. So that is what we are going to discuss in the second half of this particular meeting. Right, so there are multiple, subtypes of cell death types that occur. So, and we'll discuss most of those, if not all, right? So, yeah, with that, let us begin with this week's lecture. Uh, okay, so pick the correct statement about stem cells. The stem cells are self-renewing cells. They can only proliferate at a specific age bracket. They give rise to two daughter cells that remain as stem cells or none of the above. So in your week 8 lectures, there was a very thorough discussion about what are stem cells, right? Some new techniques that have come up or some therapeutic potential of stem cells or patient derived stem cells, right? that have been in practice now for some time, at least in clinical trials or in research laboratories. And soon we hope that they'll be used in medicine actively because they have a lot of potential. So anyway, coming back to stem cells, you have had a thorough discussion about them. So I expect you to know the answer for this one, which is option A, right? So stem cells are self-renewing cells. Yes, they are, right? The other options say that they can only proliferate at a specific age bracket. No, that is not true, right? Stem cells can proliferate whenever it is required of them to proliferate, right? So this statement is incorrect. Statement C is they give rise to two daughter cells that remain as stem cells. Right. So this is also incorrect. Stem cells, whenever they divide, they give rise to two daughter cells, one of which is a stem cell itself because it maintains its pool, stem cell pool, and the other one is a differentiated cell of some particular type. Right. So I have kept an illustration to discuss the same thing. Okay, there is just one particular participant that I have been unable to admit since the beginning of the meeting. Um, just putting it out there, because the other two I could admit. Okay. So when we talk about stem cells, as we say, they are self-renewing. That is, whenever they divide, they will also maintain their own population, right? So always after division, they will give rise to two daughter cells, one of which will be the stem cell and the other will be the transit amplifying cell or some kind of a differentiated cell type. Right. Also, it is not necessary that they will proliferate only in a specific age bracket. Okay. So moving on, which of the following statements can be used to or following factors can be used to arrest MAT alpha type of S cerebrosity cells in a G1 phase-like state, right? And your options are alpha factor, hydroxyurea, nocodazole, or none of the above. So what you have to think over here is that regarding cell cycle stages and or cell cycle checkpoints, right? And which of these particular factors will affect which particular phase in the cell cycle, right? So the answer for this one is none of the above, right? So why is it none of the above? Because for these particular yeast cells to be arrested at the G1 phase, we need to basically knock down the expression of or arrest the expression of or somehow inhibit the expression of the particular CDK cyclin that is responsible for conversion from G1 phase to S phase, right? The others that are mentioned over here, that is hydroxyurea, is basically used for arrest in the S phase, right? And nocodazole is used to arrest in the M phase, right? While over here, the most likely answer could have been CDC28TS, right? So any cyclin or CDK that is involved in G1 to S phase conversion could be, used to arrest a particular kind of yeast cell in a G1 phase, right? So the other options are not true over here. Uh, Actually, it's only now that I am actually seeing this, that alpha factor could also, arrest these cells in the G1 phase. So I think in that case, the acceptable answer should have been alpha factor, although in the first, at least time that these answers were out for last year, none of the above was the answer that was marked. But a lot of times what happens is that revisions occur, right? After the revision might have occurred, we don't have those answers. So according to me over here, alpha factor would be the correct answer instead of none of these, right? Also, this is the diagram or the representation is from a chapter in the mitotic exit network by uh, Angeles Huans, right? Okay, let me just admit the people. Now, question number 3 is which of the following can correctly define auxotrophic markers, right? So, what is auxotrophic factor or what is an auxotroph? You need to know that for this, right? Your options are genes necessary for the cells to grow under an essential nutrient deprived condition. Genes necessary for the cells to grow in the presence of drugs like hygromycin. Genes necessary for cells to grow under non-essential nutrient deprived condition or all of the above. Okay, so the answer for this particular question is the first one, option A, that is genes necessary for the cells to grow under an essential nutrient deprived condition. What does that mean? And what are the other statements? Let us go through them. The statement B says genes necessary for cells to grow in the presence of drugs like hygromycin. Now that is untrue, right? And the third statement says genes necessary for cells to grow under non-essential nutrient deprived condition, right? So it would have been either option A or option C. Now, the only difference is essential nutrient or non-essential nutrient, right? So you should know what is an essential nutrient and what is a non-essential nutrient, right? So why is it termed as non-essential nutrient? Because it is not important for this particular organism to get this nutrient as such from the environment or from the food it takes in, right? It has the machinery to basically synthesize this particular nutrient. That is why it is non-essential for it, right? Versus an essential nutrient, which will basically be a particular kind of nutrient that has to be taken up in diet by this particular organism because this particular organism does not have the machinery to actually synthesize this particular nutrient, right? So that is why it is an essential nutrient. So, what are auxiliotrophic markers? They are the genes that are necessary for cells to grow under an essential nutrient deprived condition. I have kept an illustration of so that you can actually understand what an auxotroph is, right? So, over here in this particular illustration, we are comparing an auxotroph with a prototroph, right? So in case of a prototroph, you can see that in a minimal media condition, you can see the growth of this particular organism. Even if this minimal media is supplemented with arginine, it can grow. Even when it is supplemented with lysine, it can grow, right? So this would probably mean that it does not have any, any of these nutrients, that is arginine and lysine, that it cannot itself make, even if it is given the minimal media, right, in which supplements are not provided. But in case of this arginine auxotroph, as the name suggests, it requires arginine as an essential nutrient to grow, right? In case of minimal media, which is not supplemented with arginine, you can see there is no growth. In case of lysine, supplemented minimal media also, you can see that there's no growth of this auxotroph. Only when arginine is supplemented can it grow, right? So arginine must have been an essential nutrient for this particular auxotroph, right? So it is a marker for this particular auxotroph, right? Because when present in a essential nutrient deprived condition, that is arginine deprived condition, either this 1 or 3, you can see that this particular auxotroph will not be able to grow, right? So thus this is an auxotrophic marker. Coming to question number 4. The non-pigmented plastids are called leucoplasts or chloroplasts or chromoplasts or xyloplasts? Please go through the options. Okay. So the answer for this one is leucoplasts. I'm sure that even if you did study this particular chapter pertaining to plasts in your 10 plus 2, this was mentioned over there also, right? So these were the three major kinds of plastids that we have been taught since I think class 9th or 10th, right? And we were always taught that chloroplast has this green pigment which is required for conversion of this light energy into utilizable energy, that is glucose in this particular form, right? Chromoplast, as the name suggests, is containing a chromo represents color, right? So it is a colored plastid, right? While leucoplast, even at that time we had studied, was the non-pigmented plastid. So this one does not really have a particularly defined color, right? So this is your answer, that is leucoplast. Again, there are other forms of plastids that are also present. So over here, this plastid types you can see that they are originating from one common type of cell. Now, this common type of cell is known as a protoplast. Uh, pro Proplastid, not protoplast. It is known as a proplastid or proplast cell. Okay. So what happens is that this proplast cell is kind of like a multipotent cell or a pluripotent cell which can give rise to these different kinds of plasts, right? We will call it a multipotent cell because it is only giving rise to other kinds of plastids, not any other cell type, right? So this proplast is a multipotent cell and it can give rise to different plastids such as etioplasts, which are crystalline prolamellar bodies, which are the precursors of chlorophyll, right? So chlorophyll will be formed uh, after this etioblast is formed, right? Then chromoplast is the pigment synthesis and storage unit, right? Then chloroplast is responsible for photosynthesis and lipid synthesis. Leucoplast, like we studied over here, is required for the synthesis of fatty acids and amino acids. A myeloplast is required for the storage of starch and sugars. Elioplast is responsible for storage of lipids and oils, and proteinoplast is responsible for providing sites of enzyme activity or protein activity, right? So these are the various kinds of plastids that are present. Now we are coming to question number 5, which is, in the budding yeast genome, which of the following regions have a heterochromatin-like organization, right? So I hope you remember that it was spoken of. Okay, if I would have said that, it would have given you the answer. Okay, so your options over here are the rDNA locus, the centromere, the SMC gene locus, and the telomeres. Right. So first you go through these options, you decide which one would be the right option, and then we'll discuss this. Just a moment. So the answer for this particular question is there are two answers that are actually acceptable. One is the rDNA locus and the other is the telomeres, right? So these are generally the regions in the budding yeast genome where heterochromatin-like organization is actually visible, right? So what I was saying at that point previously was that this is one of the genomes in which around the centromeres, heterochromatin is not found, right? Generally, in higher eukaryotes, that is what happens. Around the centromere, also, there is some region in which there is very dense condensation of the DNA or the chromatin, right? So, heterochromatin regions are found. Over here, this is one of the genomes wherein that does not occur, right? Apart from this, this is an illustration that is taken from this paper that was published in Epigenetics and Chromatin in 2022. Right over here, you can see that apart from the telomeric regions that you can see over here and the rDNA repeat regions, there are other regions which are the HML alpha or HML A that also we have discussed in our sessions with ma'am, right? These are other regions wherein again heterochromatin like organization is actually seen. Okay. So, coming to question number 6, your question is, in the plant system, the coaxial membrane channel that links the cytoplasm establishing cell-to-cell communication is called which one of these? Is it the middle lamella or the pectin layer or the plasma membrane or the plasmodesmata? Okay. So the answer for this one is plasmodesmata, right? If you remember the illustration of or the cartoon of a plant cell, you will see that there are pores or tube-like structures that are protruding out of one cell, and that is basically for allowing its connection to the with the cytoplasm of the neighboring cell, right? So this is what it looks like. These are plasmodesmata. So like, for example, this is one cell, this is a second cell, this is a third cell, and these are cells that are surrounding these particular cells, right? You can see for cell-cell connection or cell-cell interaction, right? And in plant cells, this happens that generally the cytoplasm is connected to one another, right? Between cells also. And this basically occurs due to the presence of this plasmodesmata, which is a pore-like component, right? It has the following other components. That is a desmotubule, a smooth endoplasmic reticulum on both ends or both the cells, and the cytosol in between, because this is basically a structure that is connecting the cytoplasm of two neighboring cells, right? And you can see that the plasma membrane lining plasmodesmata, this part, right, is connecting the two adjacent cells. So, yeah. So with that, we come to question number 7, which is the ability of the cell which has an ability to produce a complete organism is termed as totipotency or pluripotency or multipotency or unipotency. So which one of this basically could be the ability of a cell to produce an entire organism? Okay, give me a moment. Yeah. So out of these, the correct option is totipotency, right? So totipotency, this is a representation of these different way or varied kinds of potency, right? In case of totipotency, when is a cell totipotent? When it is freshly fertilized, right? And it has still not reached this particular stage wherein the embryonic stem cells are different and the outer, blastocyst, until the blastocyst stage has been reached, right? This is known as a blastocyst stage, in which this outer, red colored cells will form uh, um, the extra embryonic layers. Okay, I could not recall that name. The extra embryonic layers and this inner, embryonic stem cells are the pluripotent cells, which will form all the other cells in the body, right? Okay, let me just admit. Yeah. So as I was saying, pluripotent cells are these embryonic stem cells that will form all the other parts of the body, right? So the only difference between the totipotent cells and the pluripotent cell is that pluripotent cell can form any cell in the entire body, but it will not be capable of forming the entire organism. Why? Because it consists only of this embryonic stem cells, right? It does not contain these red colored cells, which are representing the cells of the blastocyst that form the extra embryonic layers, right? Only totipotent cells that occur before this blastocyst stage in development are the cells that can, form the entire organism. So after we basically grow up or very early in development also, we lose this totipotency, right? Only when we as egg and sperm are fertilized, that is the only time, or for a couple of days or for a few hours, that is the only time when we are or our cells are totipotent, right? After that, this totipotency is completely lost in us, and all our cells become embryonic stem cells become pluripotent. Because why are they called pluripotent? Because they can form any other cell in the body, right? Then comes multipotent. Now, what is multipotent? Multipotent is still these stem cells will still be able to form a number of different cells, but not the range is not as wide as pluripotent stem cells, right? So these embryonic stem cells that are pluripotent will probably divide into niches or cohorts of stem cells. Some have this hematopoietic fate, some have neural fate, some have mesenchymal fate, some etc. Right? And these hematopoietic stem cells can form the RBCs also. They can form the different kinds of WBCs. They can form the platelets. So there are multiple kinds of stem cells that they can form. But for example, this hematopoietic stem cells cannot form nerve cells, right? Or they cannot form skin cells, right? But although they have the capability to form a number of cells, that capability is kind of restricted to a particular cell fate, right? So that is why they are known as multipotent stem cells. Now, what will be unipotency? Unipotency will be that, for example, mm, stem cells that can only form RBCs, right? Or stem cells that can only form neurons and not glial cells, right? So these kinds of stem cells, which can still form new cells, but again, they are restricted to forming only one kind of new cell, is known as unipotency, right? So I hope with this you are also clear with what is the difference between totipotent, pluripotent, multipotent, and unipotent, right? Usually or very often, the difference between totipotent and pluripotent is asked, right? I hope you understand why pluripotent cannot form an entire organism, right? Because it does not, it lacks this particular kind of cells which are responsible for formation of extra embryonic layers during development, right? So, having said that, we'll move forward. Question number 8 is a fibroblast can be reprogrammed to cells similar to embryonic stem cells by co-expression of which of the following transcription factors? Right? Is it OCT4 or SOX2 or KLF4 or MYC or all of the above or none of the above? So you have more options to go through over here. So I would really suggest you to read the question, try to recall what you have, studied in this week. Hmm. So the answer for this one is all of the above, right? So OCT4, SOX2, KIF4, and MIG, all of them can be used. All of these are transcription factors, okay? And all of them can actually be used for different kinds of expression in the, reprogrammed embryonic stem cells, right? So, for example, I have taken these two illustrations from this particular paper in Nature Protocols by Seung Zhu et al. Right over here, the first one is a little blurry. Right, I could not get a better image of this one. You can see, let me just zoom into it, and yeah, let me just zoom into it, and you can see that over here, this is a normal MEF represents a normal fibroblast, and iPSCs represent the embryonic stem cells that have been basically derived by reprogramming this normal fibroblast, right? And you can see that there are multiple factors that are involved in this or that can be involved in this. And some of them are BMI1, MIC, KIF4, OCT4, SOX2, right? So they can all basically help in the reprogramming of this fibroblast into the embryonic or induced pluripotent stem cells, right? iPSCs. The other illustration that I have now zoomed in on represents similar kind of things, right? Over here also fibroblasts are mentioned, and there are some Yamanaka factors, right? Now, what are these Yamanaka factors? They are these OCT4, SOX2, and KIF4, right? Which can basically signal its reprogramming into stable intermediate populations. After that, if the particular or if the correct signal is given to them, these unstable intermediate populations can turn into either cardiomyocytes or neural stem cells or hepatocytes based on whichever kind of signal is provided to them. Okay, so that is that for this question. Now, your question number 9 is pertaining to neurons or nerve cells. Which of the following neurons transmit information between parts of a central nervous system? So, is it sensory neurons or motor neurons or interneurons or none of the above? Now, this one is a fairly simple question. We'll just come to the answer. The answer is interneurons, right? So, what do sensory neurons do? They basically take sensory information from the environment and send the signal to the brain. Motor neurons are performing in the exact antagonist manner. What they are doing is these neurons communicate information from the brain to the tissues and organs throughout the body, allowing for movement, right? And what are interneurons? These neurons basically make up the majority of neurons in the body. They are essentially the middleman, or they transmit information between sensory and motor neurons. They also play a key role in learning, memory, and planning. Right, so that is why the correct answer for this particular question was interneurons. Now coming to question number 10. Question number 10 says a neurotransmitter that is released in the neuromuscular junction, right, is a serotonin or acetylcholine or dopamine or glutamate. Okay. So the correct answer for this particular question is actually acetylcholine. So if you would remember, while discussing the various kinds of neurotransmitters and what is their major function, whether they are excitatory or inhibitory, or whether it is Q dependent, that whether they will act as excitatory neurotransmitters or inhibitory neurotransmitters, right? While discussing all this, ma'am did discuss about acetylcholine and how it is one of the major neurotransmitters present at the neuromuscular junction. And there are some diseases like myasthenia gravis, wherein due to unavailability of this acetylcholine at this NMJ or neuromuscular junction, there is degeneracy in the muscles that occurs, right? So, this is a representation of the neuromuscular junction. Over here, you can see this part is a muscle, right, which has this sarcolemma and the various actin myosin fibers and the, okay, the actin myosin fibers, etc. They have the A filament, Z filament, and all that is represented over here, right? And these muscles generally have nerves that signal the movement of these muscles or that transmit information from the brain to these muscles, right? So for that, at some regions in a muscle, there is this motor end plate that is present. Right? What is this motor end plate? It basically holds or hosts a nerve that is coming from the brain or the spinal cord to this particular muscle, right? And there will be a motor neuron that will be hosted in this motor end plate, and there will be synaptic vesicles that will be released in the synapse that is present over here between the motor end plate and the neuron that is coming, and that is how the information from this neuron will be conveyed to this muscle, right, through the synaptic vesicles. And these synaptic vesicles at NMJ usually contain acetylcholine, right? So this is the major neurotransmitter that is released at the neuromuscular junction. Okay, so that brings us to the end of this first part, wherein we have discussed the various, previous year questions that were pertaining to week 8 lectures. Along with that, we have briefly also gone over the major concepts for week 3, week 8, right? Uh, okay. So with that, we will basically come to the second part of today's presentation or today's lecture, which is going to be the different types of cell deaths, right? So what we'll discuss is cell death types apart from apoptosis and necrosis. Like there is autophagy that occurs, right? Apart from that, also there are other kinds of cell deaths that occur. Some of which I had, mentioned the last time we spoke. Some of them could be ferroptosis, pyroptosis, necrosis we have already counted as a major one. There are multiple of them, right? So that is what we are going to discuss. And the major part for this particular discussion, I have picked up from a review paper that I have cited, right? You can go and read that review paper, although I have covered it entirely in this particular presentation. Also, for more, detail about it, since I usually like to, refer to books and to teach via what is given in the books, because the writing over there is very, very simple and easy to understand and easy to convey. But the different kinds of cell deaths that have come up in the past 10 years or so are so very many that they have not really been included in these books as of now, right? So it will be better if you want to learn about them, either separately go and type and find Wikipedia pages and look at them, or find more review papers and you can actually go through what these different kinds of, cell death types are. So we'll begin with this lecture. Just give me a short water break. Yeah. So let us begin with this particular presentation also. When we speak about cell death mechanisms in eukaryotes, there are very many mechanisms that are present. Okay. Like the organisms, they constitute, the cells also die in different ways, right? So, if you talk about humans in general, there are a lot of kinds of death that can occur, right? It could be death due to some brain disease, it could be death due to stroke, due to a heart attack, due to heart failure, or due to cancer. So there are different kinds of deaths that are occurring, right? So even at cellular level, there are different kinds of deaths that can occur. The death can be predetermined, programmed, and cleanly executed, as in the case of apoptosis, or it can be traumatic, inflammatory, and sudden, as many types of necrosis exemplify, right? So, apoptosis, when we speak about it, is coordinated by death ligands, right? We have studied about that also in the previous week. And caspases and B cell lymphoma 2 or BCL2 family proteins and their downstream effectors. Events that can lead to apoptosis include mitotic catastrophe and anoikis, right? So, mitotic catastrophe will basically mean that, for example, a cell that is failing to undergo mitosis, it should undergo mitosis, but it is failing to undergo mitosis, or it has basically failed to go through with mitosis because of the failure. Because of some maybe DNA or chromatin morphology that was different, and some checkpoint was present which did not allow this mitosis to progress, etc. Right? So there could be multiple kinds of mitotic catastrophe events that could occur. And they could lead to apoptosis. The other one that is mentioned over here is anoikis, right? What is this anoikis? Anoikis basically means that for cells to be healthy, they have to be present in a fixed location, and they need to have some intercellular contact or intercellular connections, right? So sometimes what happens is that if a cell will detach from its neighboring cells, and it will detach from the extracellular matrix that it was present in, and it will kind of wander around, that phase is known as anoikis. Now, this cell, because it has lost its cell-cell connections and its connection to the ECM, now it will be apoptosed, or it will die due to some other cell death mechanism, right? So, because this is not what should occur, right? It is abnormal, so it should not occur. So, these events, such as mitotic catastrophe and anoikis, don't just lead to apoptosis, they could lead to other kinds of cell death. We'll look at that also. When we talk about necrosis, this has been considered an abrupt and uncoordinated cell death, right? But despite that, it has many molecular events that are associated with it. Okay. Autophagy, which is the third major kind of cell death that is widely acknowledged, is generally a catabolic pathway that operates to ensure cell survival. It can also kill the cell through mechanisms such as autosis. Now we will study what this autosis is, right? Other cell death mechanisms include endosis, ferroptosis, lysosome-dependent cell death, parthenoatose, etc. Right? We'll discuss all of these. So, first, what is apoptosis? Right? Apoptosis literally has been derived from a Greek word, and it literally means falling off. Right? So, for the this phenomena of, or this mechanism of something falling off from the trees is also predetermined, right? There are seasons wherein the tree will be full of leaves. There are seasons wherein the leaves will just fall off, right? So that is also a programmed mechanism. So apoptosis is a programmed cell death, or PCD, that plays a crucial role in development and in diseases. Okay. The morphological manifestations of apoptosis include cell membrane blebbing, shrinkage of the cell, and formation of apoptotic bodies. Now, what is cell membrane blebbing? It means that if the cell membrane is not smooth anymore, there are, kind of cracks that are coming on it, right? So blebs are coming. I will show it when I show the representation for apoptosis. Then shrinkage of cell occur, right? So now this is important because there are some kinds of cell death mechanisms which involve swelling of the cells. There are some others wherein there is condensation or shrinkage of the cell. So apoptosis is one of them, right? Then there is also formation of apoptotic bodies. Now, what are these apoptotic bodies? That whatever fragments of the cell organelles have occurred, or fragmentation of DNA has occurred, is already within the cell. Right now, how it will apoptosis, that it will basically tear away from the cell while maintaining parts of plasma membrane intact around it. Now, how does it help? It helps because when such kind of a mechanism occurs, it does not signal inflammation or inflammatory molecules, right? So when inflammatory molecules will not be signaled, there will be no harm to other cells that are occurring in its periphery, right? Only the uh, macrophages are triggered so that they will come and they will eat whatever apoptotic bodies are present, right? So apoptosis can generally be executed via three different pathways. One is the extrinsic pathway, then comes the intrinsic pathway, and then the endoplasmic reticulum stress or ER stress induced pathway. Right. So we did discuss the extrinsic and the intrinsic pathway last time in the last session. The third one is ER stress induced pathway. Now, these pathways involve the action of cysteine proteases, which are known as caspases, that cleave their target proteins after an aspartic acid residue. Right. So, where was I? Yeah. So, these pathways, all of these extrinsic, intrinsic, or ER stress-induced pathway, all of them basically involve action of cysteine proteases, which are known as caspases, which cleave their target proteins after the aspartic acid residues. Okay. So, any protease will have a specific site where it can cleave the protein, right? So, over here, the site is after the aspartic acid residue. So these are the pathways that we talked about. I will discuss about them in detail in the coming slides. Let us just go through them in an overview so that when I discuss about them in that, text heavy slides, you'll be able to follow. Okay, so over here we are saying that the end result of these pathways is apoptosis, right? Whether it is a ER stress regulated pathway or the intrinsic or the extrinsic pathway, right? So what will be an extrinsic pathway? That there is some mechanism. Just a moment. There's somebody who's still trying to join. I'll just share my screen again. Yeah. So in the extrinsic pathway, what happens is that there are these death ligands, right? What are the death ligands? That they are some molecules that are signaling for the cell to die, right? These molecules could be molecules like TNF alpha, that is tumor necrosis factor alpha, TRAIL, or FASL, right? And then there are death receptors, which are receptors for these, death ligands. So they are FASR, TNFR1, Trail R1, or Trail R2, right? And these death receptors are basically, when they interact with these death ligands, they uh, stimulate a signaling cascade, right? This signaling cascade involves the formation of this disc, which is basically a destruction complex, which will basically activate the procaspase 8 and procaspase 10. Once they are activated, they will be converted into their active form, that is caspase 8 and caspase 10. These are the initiator caspases, right? After they are activated, they will activate the executioner caspases, which are procaspases 3, 6, and 7. They will basically be converted to their active form, that is caspase 3, caspase 6, and caspase 7. And these are executioner caspases, and they will lead to apoptosis to occur. Okay, and this apoptosis will also be through the intrinsic pathway, or it could be independent of the intrinsic pathway directly. Caspase 3 is capable enough to give this signal for apoptosis to occur, right? So out of these executioner caspases, also caspase 3 is the most relevant one. Now, what happens in an intrinsic pathway? In an intrinsic pathway, uh, not because of some extracellular signal, but because of the signal from within the cell, that is usually from within the mitochondria, occurs, right? It could occur due to stress or excessive ROS that is present, or DNA damage that has occurred, right? So, in response to these, what could happen is that the mitochondria could release cytochrome C, which is present in the inner intermembrane space, or intermitochondrial membrane between inner and outer membrane. This particular protein is present, right? Cytochrome C. Now, what could happen when this cell faces stress or DNA damage or excessive ROS? It could happen that there are pores that are formed on the outer mitochondrial membrane. We have studied how this pore formation occurs, right? That the BCL2 family proteins, which are anti-apoptotic, they will probably be, silenced, and then backs will be recruited to this outer mitochondrial membrane. They will, initiate the recruitment of even more backs, and they will form this mitochondrial pore, uh, through which the cytochrome C can be released into the cytosol. Right? Once it is released into the cytosol, it can basically activate the procaspase 9 into active form caspase 9. This again is an initiator caspase, which will go and activate caspase 3, 6, and 7, which are the executioner caspases, which will form or which will cause apoptosis to occur. Right? Then comes the third kind of pathway, that is the ER stress induced pathway. Right. In this, IRE1 is one important protein, right? We will talk about this. What happens is that this protein, if there is some stress in the endoplasmic reticulum, or the protein folding is not occurring properly, then it will probably halt the protein synthesis also for that time. It will try to correct it. If it is unable to correct it, this IRE1 will basically signal caspase 12 activation, and once this caspase 12 activation is signaled, it can lead to apoptosis, right? Or through GNK mediated pathway, again, it can lead to mitochondrial mediated caspase 3 activation, which will again lead to apoptosis, right? So this particular GNK mediated pathway will also lead to release of caspase 3 and apoptosis. Now we have discussed them in this representation. Now let us read about these three pathways, right? So extrinsic pathway of apoptosis, like we have already discussed, the extrinsic pathway is triggered by the binding of ligands to the transmembrane to the transmembrane death receptors, such as tumor necrosis factor receptor 1, TNFR1, or FAST receptor. Right? The ligand binding causes clustering and cytoplasmic domains of these receptors, which in turn recruits adapt proteins containing corresponding death domains. right? These death domains of these adapter proteins serve as binding site for different upstream procaspases, such as caspase 8 and 10, to form death-inducing signaling complex, that is DISC, right? Once this disc is formed, it will activate these caspases, that is caspase 8 and 10. The activated upstream caspases then activate the downstream executioner caspases, which are caspase 3, 6, and 7, which will execute the cell death or the occurring of apoptosis. Right? When we talk about intrinsic pathway of apoptosis, it is also known as the mitochondrial pathway. Right? It involves the release of cytochrome C from the mitochondria to the cytoplasm in response to cellular stress. The cytochrome C, in association with apoptotic protein activating factor 1 or APAP1, forms an apoptosome. Within apoptosome, procaspase 9 is activated to become caspase 9. Caspase 9 in turn activates the downstream effector caspases. Right? Again, these effector caspases are caspase 3, 6, and 7. Right? The mitochondria further release pro-apoptotic signal proteins such as apoptosis-inducing factor AIF and endonuclease G. AIF translocates to the nucleus and facilitates DNA fragmentation and chromatin condensation. After this, stage 1, chromatin condensation, which is also known as ring condensation, it ensues the translocation of endonuclease G to nucleus, leading to further fragmentation of the DNA. Right? Finally, after this, caspase activated DNA, that is CAD, is translocated to the nucleus, leading to stage 2 condensation or the necklace condensation. After the stage 2 condensation happens, the mitochondrial phase of apoptosis is regulated by BCL2 family proteins, right? The pro-apoptotic proteins of this family, such as BAX and BAC, create pores on the mitochondrial membrane in response to apoptotic signal, which facilitates the release of cytochrome C. Meanwhile, other apoptotic proteins such as BIM keep anti-apoptotic protein, that is BCL2, inactive. The release cytochrome C through its interaction with APAF1 activates caspase 9. Meanwhile, mitochondria released a second molecule, which is aptly named as second mitochondrial derived factor activator of caspase, that inhibits IAP or inhibitor of apoptosis proteins, preventing the latter from deactivating caspase 9. Now, if caspase 9 is not deactivated, it is fully enabled, it will carry out the death by activating downstream caspases such as caspase 3, right, which is an executional caspase, and it will lead to cell death. Now, the third pathway was the ER stress-induced pathway of apoptosis. There are certain infections or hypoxia conditions or starvation or chemicals such as tunicamycin or thapsigargan or brefeldin A and non-homeostatic changes in the secretory functions of the ER contribute to the accumulation of unfolded or misfolded proteins in its facilitating ER stress-induced cell death. Okay. When misfolding of proteins occur in ER, unfolded protein response pathway is initiated. Now, what does this unfolded protein response pathway do? It halts the global protein synthesis and focuses specifically on replacement of unfolded proteins with correctly folded proteins. There are three major components of this pathway, which are protein kinase, RNA like endoplasmic reticulum kinase, that is PERK, inositol requiring enzyme 1, IRE1, and activating transcription factor 6 or ATF6, which work in tandem to restore proteostasis inside the ER. Right? But what happens if the cell's attempts to resolve the persistence of unfolded proteins fails? It chooses to initiate apoptosis instead. IRE1 plays a significant role in apoptosis induction via a caspase 12 mediated pathway and also via TNF receptor associated factor 2 apoptosis signal regulating kinase 1, that is TRAF2 ASK1 complex mediated JNK signaling and P38 MAP kinase signaling cascade. Right. Now, what will this GNK pathway activation do? It will lead to apoptosis by inhibition of anti-apoptotic protein BCL2 and activation of pro-apoptotic protein BIM1. Sorry, BIM. The P38 MAP kinase pathway facilitates the cell death via activation of key protein locating downstream of the PERK and the ATF6 pathways called the CEPP homologous protein or CHOP. Right? The CHOP facilitates the expression of pro-apoptotic proteins such as caspase-3 activator triples, homolog 3, that is TRP3, growth arrest, and DNA damage protein 34. The tumor necrosis factor receptor protein, the death receptor 5, promoting caspase-8 dependent apoptosis. Further, CHOP inhibits BCL2 transcription and promotes expression of the BIM. So, in short, we have already discussed this earlier, right? When we went through that signaling pathway. The stress induced apoptosis has been reported in diseases such as Alzheimer's and Parkinson's disease, retinitis pigmentosa, glaucoma, macular degeneration, inflammatory diseases, tumorigenesis, and metabolic diseases. There are some drugs such as Vithafirin A that can induce stress-mediated apoptosis via activation of caspase-4, a functional homologue of caspase-12. Then BAX inhibitor 1 or BI1 or TIMBIM6 and BCL2 or BCLXL are major inhibitors of the stress-induced apoptosis. Right? Uh, in correspondence to this, I also wanted to go through this particular pathway, which is perforin granzyme pathway. I am sure you might have heard of it when you would have been studying about how some viral particles or some bacterial particles are basically infecting us and how they are getting through into our cells, and then what our cell is doing in response to that, right? So what happens, or when this pathway comes into picture is, it addresses infections, right? For addressing infections, the cell executes a pathway that bears a resemblance to the apoptosis pathway, right? It is very similar to apoptosis. It is known as the perforin granzyme pathway. Now, cytotoxic T lymphocytes and natural killer cells, for example, directly contact pathogen infected and transformed cells and eliminate them without harming without harming the non-infected cells. Right? So that is the purpose of apoptosis also, right? That if it will not induce inflammation, the non-harmful cells or the normal healthy cells will not be, harmed. In this mode of cell death induction, perforin, which is a membrane disrupting protein, and a specialized group of serine proteases, which are known as granzymes, are exocytosed from the immune cells to initiate apoptosis in the infected cells. Right? So, what will happen? This granzyme will facilitate the caspase independent pathway via Fas associated DNase complex called SET, leading to DNA fragmentation. Right? So Fas was one of the proteins that was present in the death receptor, is also a death ligand, and it initiates that disc formation, right? But over here, it will do it in a caspase independent way, right? This pathway is insensitive to BCL2 protein. So whether this BCL2 is present, it will not be affected. We know that BCL2 protein is anti-apoptotic, and there are some BCL2 family proteins like back and backs which are pro-apoptotic. But this pathway is basically insensitive to these proteins and is independent of the membrane integrity of the mitochondria, right? Even if it is porous or non-porous, it will not be affected. Then there is another granzyme, which is granzyme B, which on the other hand works via caspase 9 and caspase 3, that is the mitochondrial pathway, right? Or the intrinsic apoptotic pathway. Elevates reactive oxygen species levels to promote the cell death, right? So what happens in this perforin granzyme pathway is that these natural killer cells, they will release the granzymes and perforins in the presence of perforins, which will form, pores in the membrane. These granzymes will get into the cell. Granzyme A will follow a caspase-independent pathway and lead to DNA fragmentation and apoptosis. Granzyme B, on the other hand, will increase the presence of ROS, which might induce the intrinsic pathway, and it will lead to activation of caspase 9 and caspase 3, which will again lead to apoptosis. Okay, then let us study what are the major characteristics of apoptosis, right? A prominent feature of apoptosis is the absence of a systematic or localized damage to other cells owing to lack of inflammation during its initiation, completion, and clearance of the dead cells. The apoptotic cell achieves this by carefully wrapping the cell membrane around the now fragmented cell, forming apoptotic bodies. Okay, so this was a cell. It will undergo on some level condensation and shrinkage, right? After this, membrane blebbing will occur, right? Membrane blebbing is this form of membrane that occurs, right? After this, what will happen is that apoptotic bodies will be formed, and these lysed DNA and lysed intracellular compartments will actually be packed into these small apoptotic bodies, and then they will be engulfed by macrophages, right? That is how apoptosis will occur. Then, when the nucleotides such as adenosine 5-triphosphate or ATP and uridine 5-triphosphate or UTP, which are released by apoptotic cells, serve as the find me signal for the macrophages. Right. So first, let us look at this top view of this illustration or this cartoon. Right. So what happens is that if there is a cell, right, if it will have to give a find me signal to the to the macrophages, what it will do is that it will basically represent this ATP and UTP on itself, right? So ATP and UTP should ideally be inside the cell, and that too inside the mitochondria mostly, right? If it is present on the cell, right, it will serve as a signal for macrophages to come and find that okay, there is something wrong with the cell, right? There is another one, that is CX3Cl, which is a inflammatory ligand only that is present, which could also basically serve as a pro or a subsidiary receptor for this particular macrophage to come and find this particular cell, right? So these will act as signals. Once a macrophage gains sufficient proximity, once it has sufficiently come close to this cell that has to undergo apoptosis, the apoptotic bodies enable the former to engulf them by displaying the phosphatidylserine or the eat me signal on their surface, right? So if a cell, for example, is healthy, it will not have any phosphatidylserine on its surface, right? It will have normal cell surface receptors. But if this particular one is an apoptotic cell, right? It has already blebbed and formed apoptotic bodies. On these apoptotic bodies will be phosphatidylserine, right? Now, this phosphatidylserine is generally or normally present in the inner side of a normal cell, right? When it is expressed on the outer side, it will give a eat me signal, and this phagocyte site will definitely know that okay, this is one cell that has to be removed, or this is one apoptotic body that has to be removed, and it will just engulf it, right? Then the process of engulfment of apoptotic cells by macrophages is called efferocytosis. It is a process bearing similarity to the micropanocytosis. Okay, if an apoptotic cell fails to undergo proper clearance via efferocytosis, it may undergo secondary necrosis. Inefficient efferocytosis can lead to diseases such as cystic fibrosis or rheumatoid arthritis. So, this particular, uh, you know, phenomena of engulfing of this cellular debris or apoptotic cell bodies by the phagocyte is known as efferocytosis. Okay. Then we come to necrosis, which is the second major kind of cell death. Necrosis is classified as an inflammatory form of cell death, which is characterized by the rupture of cell membrane. Okay, so infection, high dose radiation, electric, chemical, or poison shock, high pressure, and asphyxiation can cause necrosis. During necrosis, the cell membrane integrity is broken down and the cytoplasmic constituents ooze out into the extracellular space. Right? This is one of the major differences between necrosis and apoptosis. In this, cell membrane is broken down. Over there, cell membrane is still intact, right? And it is even when apoptotic bodies are formed. Unlike apoptosis, necrosis does not follow an ordered signal transduction pathway, right? So over here, there is no particular pathway that has to be followed for a cell to undergo necrosis, right? It could be a sudden traumatic process also. But despite that, there are some molecular signals that we know are there when necrosis is occurring, right? So let us read about them. There are some cues for necrosis to occur. It must be noted that necrosis need not be confined to cells that are integrated into tissues. Cells in vitro culture medium can also undergo necrosis, right? It can very well be seen. For example, ATP depletion has been shown to induce necrosis in primary cell cultured in vitro, right? Similarly, there are some drugs such as tamoxifen, which at high concentration can induce necrosis in MCF7 and HL60 cells. Right? Now, what is interesting to note over here is that the same drug, tamoxifen, at lower doses will promote apoptosis. Right? And at minimal dose, it will induce autophagy. So this means that there is a switch between these cell death pathways that occurs, right? So based on concentration of something that is given also, it might, vary whether the cell is undergoing autophagy or apoptosis or necrosis, right? Then arsenic trioxide is another example of necrosis inducing drugs. Okay. Then other cues for necrosis to occur include that although there are pivotal switches that decide between apoptosis and necrosis, they are yet, not fully identified. There are elevated levels of intracellular loss, inhibition of caspase activity, and reduced levels of ATP are known to trigger this form of cell death. Right. Then DNA strand break caused by ROS, glutamic excitotoxicity, can promote excessive activation of DNA repair enzyme poly ADP ribose polymerase 1 or PARP1, leading to ATP depletion and thereby necrotic cell death. Similarly, an increase in calcium concentration can lead to leaky mitochondria that further reduces ATP levels. When apoptosis is blocked by caspase inhibition, the mode of cell death can shift to necrosis. There are certain cell surface receptors such as RIG1-like receptors or RLR, toll-like receptors or TLRs, nucleotide oligomerization domains, NOD-like receptors, NLRs, are involved in pathogen recognition and innate immunity activation. Right? TLRs you might remember, and NLRs also a lot of you might remember that they help in innate immunity. Right? They can induce necrotic cell death by identifying double-stranded RNA, liposaccharides, or flagellin found in pathogens. Further, synthetic double-stranded RNAs on recognition by TLRs can induce necrosis. The persistent opening of high conductance permeability transition pores of mitochondria called mitochondrial permeability transition or MPT can also drive a cell to necrosis. Now, MPT-driven necrosis involves a rapid increase in permeability of mitochondrial membrane induced by elevated levels of intramitochondrial calcium and oxidative stress. Now, if you remember, when we talk about apoptosis, we said that in that also there is a change in the mitochondrial membrane permeability, right? So it must be noted that mitochondrial rupture releases pro-apoptotic factors such as cytochrome C that can induce apoptotic cell death as well. However, the rapid loss of ATP can tilt the cell's destiny to MPT-driven necrosis. This is an illustration of necrosis. Over here, there is a normal cell, and due to some stress, heat, trauma, anoxia, and pathogens, what happens is that the ROS and calcium levels can increase, and there could be a depletion in the ATP levels that are present. This could lead to mitochondrial damage and increase in permeability of these mitochondria, and it can also lead to lysis of ER, swelling of mitochondria, and release of the cell contents, right? And the cell membrane is also not not the integrity is not maintained. It has broken down. It has lysed, and all these contents are basically being released outside of the cell. Then there are multiple other cell deaths that are similar to necrosis. While necrosis is an unprogrammed form of cell death, there are related cell deaths with cell membrane rupture as a common denominator, right? That is why we are saying that they are similar to necrosis, because they also have cell rupture of cell membrane that occurs. These include methuosis, necroptosis, netosis, pyronecrosis, and pyroptosis. Such necrosis-like deaths are nevertheless regulated by different signaling pathways and external or internal inducers and mediators, right? So it might be possible that among these, the how it is occurring is same between two of them, but what is actually causing this death to occur is different. That is why they have been different, given different names, right? Or it might happen that out of these maybe 5 or 6 types of cell deaths, um, the causative molecule is the same, but how it is being affected and how the cell death is occurring is different, right? So there could be differences at different levels that could occur. Okay. Sorry. So let us go through the first one, which is methuosis, right? Methuosis is characterized by accumulation of fluid-filled cytoplasmic vacuoles in the cell and eventual detachment of the cell from the neighboring cells. Right, so over here there are fluid-filled vacuoles that are present, and it will lead to detachment of this particular cell from the neighboring cells, which is also what anoikis is. Okay. This RAS-induced caspase-independent cell death was first observed in malignant glioma and gastric cancer cell lines, right? So it is caspase-independent cell death, so it is definitely not like apoptosis, it is more like necrosis. Unlike the autophagosomes associated with autophagy, vacuoles in methuosis are bound by single membrane and derived from macropinosomes, right? So over here, in autophagosomes, in autophagy, what happens? There is a double membrane bound structures. Over here, it is single membrane bound, right? That is why it is different from autophagy also. The vacuolization can lead eventually to the rupture of the cell membrane. At molecular level, the process of methuosis involves the induction of RAS-GTP pathway that is independent of the RAS-associated proliferative pathway. Okay. Now, compounds such as chalcones and their derivatives have been shown to promote methuosis via disruption of vesicular trafficking and procathepsin processing. Similarly, an anti-cancer agent, silmitarcept, tarsortem, can induce methuosis in cholangiocarcinoma and colorectal cancer cell lines. Right? If it is basically inducing some form of cell death mechanism in a cancer cell, it is good, right? Because cancer cells are also going to die because of that. So interestingly, the same drug induced autophagic cell death in pancreatic cells and apoptosis in non-small cell lung cancer cells. Right. So now over here, the drug being used is the same. I don't know whether the concentration that is used is the same or not, but it can induce different forms of cell death in different kinds of cancer cell lines. Right. So that is what is written over here. Now, what is methosis? We have already gone through it, that there is a presence of non accumulation of non-acidic large fluid-filled vacuoles, right? Due to this, there are some pathways that go up, and it leads to eventually leads to cell death, right? Okay. Then comes necroptosis, right? So this kind of sounds like a mixture of necrosis and apoptosis, right? So upon pathogenic stimulation or in presence of drugs such as chikungunya, a TNFR1 associated receptor interacting protein kinase called receptor interacting protein kinase 1 or RIPK1 induces necroptosis, a type of cell death whose features bear resemblance both to apoptosis and necrosis. Binding of the ligand to a TNFR1 releases the formation of complex at the cytosolic site of TNFR1 that contains proteins such as RIPK, TRAF2 or TRAF5 and tumor necrosis factor type 1 associated death domain or TRAD. Right, so over here there is a death ligand of some sort that is present. That is how it is similar to apoptosis. From this point, the cell may choose one of the three fates. What are the three fates that can be chosen? If ubiquitination of this RIPK1 is occurring, then it is mediated by cellular inhibition of apoptosis proteins and TRAF2 or 5 prevails, cell death is aborted, right? If this particular protein is ubiquitinated, cell death will be aborted. If inhibition of CIAP is in concert with deubiquitination of RIPK1 prions, caspase 8 activation ensues, resulting in apoptosis. So apoptosis could also occur depending on what is being activated. Over here, caspase 8 will get activated. However, when there caspase 8 activity is inhibited at this point, two other proteins that is receptor interacting protein kinase 3 and RIPK1 form the necrosome complex, which then phosphorylates mixed lineage kinase domain like protein, MLKL. Okay. Now, after this happens, the phosphorylated MLKL then substantially increases mitochondria-dependent ROS production. Further, MLKL translocates to plasma membrane to disrupt the membrane integrity, leading to cell death. Right. So this is its feature that is resembling necrosis, that the cell membrane integrity is disrupted. Unlike apoptosis, necroptosis induces inflammation, which is seen prevalently in neurodegenerative diseases. In further contrast to apoptosis, necroptotic cell death have randomly degraded DNA. Okay. On a therapeutic perspective, it has been shown that cancer metastasis can be restricted through induction of necroptosis. RIPK1 and MLKL inhibitors such as necrostatin 1 can block this necroptotic cell death. Now, this is an illustration of necroptosis. Like we have talked, there is a ligand which comes and binds to the TNFR1 receptor on the plasma membrane. It could lead to formation of a complex or a destruction complex like TRAD or RIPK1 and associated proteins. It could lead to the inhibition of caspase 8 and the formation of a necrosome complex which contains RIPK1 and RIPK3, right? This will induce MLKL formation, and this MLKL will cause increase in ROS, decrease in ATP, and it will also lead to necroptosis, and this MLKL phosphorylated complex will go and it will also disrupt the plasma membrane. Okay. Coming to netosis, right? Now, what is this netosis? It basically occurs in neutrophils, right? And it is a defense against infectious pathogens, right? Now, what is NETs? When encountering pathogens, neutrophils release neutrophil extracellular traps, which are known as NETs, right? Consisting mostly of DNA fibers to capture and kill the pathogens, right? So what will happen? This NADPH oxidase dependent cell death progresses with a substantial elevation of ROS, leading to activation of arginine deaminase called protein arginine deaminase 4 or PAD4. This PAD4 then citrullinates, which is it converts arginine into citrulline via deamination in the histones, right? The citrullination occurs in histones, and this facilitates decondensation of the nuclear chromatin. After this, besides enzymes such as elastase and myeloperoxidase, which are released by neutrophil granules, translocate to the nucleus, facilitating the unfolding of the chromatin. After this, the disruption of nuclear envelope follows, releasing the chromatin into the cytosol. Onto this, decondensed chromatin, many cytosolic proteins are added. Finally, the outer membrane of the neutrophil disintegrates and the net is released. Mast cells and eosinophils also show netosis and is termed as etosis, right? Because a neutrophil, it does not include neutrophils, that is why it is known as etosis. Now, what happens in netosis? This neutrophil is activated by pathogens. It leads to decondensation of the chromatin, and through PAD4 activation, it leads to the release of this nets, that is neutrophil extracellular traps, which will basically go and trap these pathogens that are present outside, and it will basically lead to cell death, right? But this cell death is for a good cause because it is trapping the pathogens, and it is basically ultimately saving our body, right? But again, cell membrane disintegration is occurring, so it is similar to necrosis. Then comes pyronecrosis. Now, pyronecrosis is a necrosis-like cell death stimulated by pathogens such as Shigella flexneri, right? It operates chiefly through NLR family pyrin domain containing 3 NLRP3 receptor, which induces the formation of inflammasome complex. Specifically, upon stimulation by molecules of the pathogen, NLRP3 oligomerizes and induces oligomerization of another protein, which is ASC, that is for apoptosis associated spike-like protein containing caspase recruitment domain, which creates an inflammasome complex. The complex induces the production of interleukin I beta. Further, a pro-inflammatory factor, HMG-B1, is released. Finally, the cell membrane ruptures, releasing intracellular components. So that is what occurs in pyronecrosis. Now, at this point, I would like to point out that we are discussing in every kind of cell death type, or in every kind of cell death, we are discussing some major factor that is different from some other major factor in some other type of cell death, right? For example, over here, it would be HMG-B1, right? In the previous one, when we were discussing this netosis, it was this PAD4, right? Before that, in necroptosis, it was this RIPK1 and MLKL, right? So these factors, which are unique to these different kinds of these factors, which are unique to these different kinds of cell death mechanisms, are actually used to identify whether netosis is occurring or pyroptosis is occurring, or some ferroptosis, some other cell death mechanism is occurring, right? That is what will signal this. So every time you will not be able to see what is happening, right? But you can detect it via maybe fluorescence staining through antibodies, and then looking through a microscope, or by tagging these particular proteins by looking for their transcript levels, for their protein levels, etc. Right? That is how these varied kinds of cell death mechanisms are actually identified. Then it also may be noted that cells that have already initiated apoptosis will not release this HMG-B1 even when they initiate secondary necrosis later. Okay. So in pyronecrosis, we saw that in case of pathogens that are present, there will be an inflammasome complex that will be formed by the presence of this NLRP3 and ASC that is present, and this will basically promote the pro-inflammatory IL beta, which will lead to pro-inflammatory cytokines, and it will also simultaneously form the death of this particular cell that is actually releasing it, right? Then we come to pyroptosis. Now, what is pyroptosis? It is a form of regulated necrosis which is stimulated by factors such as microbial infection. It plays a major role in immune defense against infections. Nevertheless, other factors such as stroke can also lead to pyroptosis. What happens in this? When a cell is damaged, the biomolecules such as damage associated protein molecular patterns, DAMPs, initiate the inflammatory response by binding to DAMP receptors such as TLRs. Right? After this, with infections, microbial molecules having PAMPs or the recognition sites initiate the response by binding to respective pattern recognition receptors. Right? The inflammasome complex, consisting of sensory proteins ASC and procaspase-1, triggers inflammation via activation of caspase-1 and subsequent activation of gasdermin D. This gasdermin D makes spores on plasma membrane. Inflammatory cytokines also play a significant role in assisting pyroptosis. The cell in this case swells and it ultimately ruptures. In case of apoptosis, the cell shrinkage used to occur. Over here, the cell will swell and it will ultimately rupture its membrane, resulting in release of pro-inflammatory contents, right? That is how pro-inflammatory contents will be released. So this is a representation of pyroptosis, wherein the PAMs that are recognized by PRRs or pattern recognition receptors will lead to the formation of inflammasomes similar to pyronecrosis, right? And after this inflammasome is formed, it will lead to activation of caspase-1, which will also lead to formation of cleavage of gasdermin D end domain, which will release pro-inflammatory factors. Along with the release of these pro-inflammatory factors, like how will the release of this pro-inflammatory factor occur? It will basically cause disruption in the cell membrane, and now this cell membrane will be broken at places through which inflammatory cytokines will be released or pro-inflammatory factors will be released, right? And the cell will subsequently die because now there is no integral cell membrane that is actually bound monitoring the cell, right? Then there are other forms of cell deaths that are also present. First, we'll begin with autophagy, right? Because this, again, we said was one of the major forms of cell death. It is the natural conserved degradation of the cells that removes unnecessary or dysfunctional components through a lysosome-dependent regulated mechanisms. It allows the orderly degradation and recycling of cellular components, although initially characterized as primordial degradation pathway induced to protect against starvation. Right. Initially, that was how autophagy was described, that only starved cells are removed by autophagy to basically so that other cells also don't starve. It has now become increasingly clear that autophagy also plays a major role in homeostasis of non-starved cells. Defects in autophagy have been linked to various human diseases, including neurodegeneration and cancer, and interest in modulating autophagy as potential treatment for these diseases has also grown rapidly, right? So over here, not going much into detail, this is an illustration that I have picked from this particular paper in Cell Discovery. Uh, Volume 6, right? Over here, you can see that even autophagy is of multiple types, right? There are macro autophagy, there is micro autophagy, depending on what is it that is being autophagocytosed, right? Whether it is a small unit, whether it is a big organism, whether it is an entire cell organelle or just a part of that cell organelle or just a protein, it will be, there are different kinds of autophagies that are present, right? Along with this, there are also chaperone-mediated autophagy or and RN or DN autophagy, wherein either if a protein is being phagocytosed, then it is occurring due to the presence of some chaperone that is leading it to this particular place that is allowing its autophagocytosis to occur, right? Or nucleic acids are being basically autophagocytosed. So there are different kinds of autophagies that are present. Now, along with this autophagy that we largely already knew about, there are other cell death types that we might not know about, right? One of which is autosis, right? Now, what is autosis? It is basically also a form of autophagy associated cell death. Autosis is a term that is used for the first time in 2015 by Louis and Levine, right? So it is fairly recent, not even a decade ago, right? It describes, they described it as an autophagy gene-dependent non-apoptotic cell death with distinct morphological features. A viral that was that for which meant activator of transcription, a cell penetrating peptide was fused to a small peptide sequence, Becklin 1, right? That is that Becklin 1 was formed. And it was found to induce this particular form of cell death, which is known as autosis. Now, in autosis, plasma membrane gets ruptured at focal points. Mild condensation of chromatin and presence of abundant autolysosomes and autophagosomes and their disappearance at the later stages, swollen enrichment, electron-rich mitochondria, and fragmented swollen ER are additional indicators of autosis. This form of cell death depends upon Na plus K plus ATPase, right? Or this particular sodium potassium pump. Cerebral hypoxia, hypoxic ischemia, and hepatocyte injury associated with several severe anorexia nervosa are also known to promote autosis, right? So any condition which basically restricts the oxygen that is being supplied to a particular region will induce autosis. Pharmacological agents such as cardiac glycosides, such as oxygen, digoxin, digitoxygenin, etc., that inhibit Na plus K plus ATPase and knock down alpha subunit of these sodium potassium pumps, inhibit autosis, confirming the role of this pump in its initiation and progression. Right. So probably the function of this pump is what we will use as a marker for autosis. Now, there was a study that also showed that ginger extract induced autosis in cultured pancreatic cancer cells as indicated by increase in the LC3 2 and to LC3-1 ratio, right, which is an indicator of formation and prevalence of autophagosomes and the vacuolization of cytoplasms. The phycocyanin is one of the major pigment which constitutes spirulina, and it can also induce autotic cell death in pancreatic 1 cells. Also, so if from this what we can see is that we can use ginger extract or phycocyanin or any such autosis inducing agent to actually stop growth in cancer cells, right? It could be used as a therapy probably in future. Then drugs such as nerifolin, digoxin, digitoxygen can inhibit us digitoxygenin can inhibit autosis, right? So instead of inducing autosis, these drugs can actually inhibit it. Now, this is a representation of autosis. The major thing you want to remember in this is that hypoxic condition, ischemia, starvation, and autophagy can actually induce peptides that cause autosis, and the activity of Na plus K plus ATPase pump will also be important in the induction of autosis, right? The presentation of autosis will include fragmented endoplasmic reticulum, presence of autolysosomes, dilated ER, focal plasma membrane ruptures at various points, focal bloating of perinuclear space, that this perinuclear space has basically increased. Then chromatin condensation, mitochondria might, look like electron dense swollen mitochondria. Then nuclear membrane convolutions might also occur, right? And all these will eventually lead to autotic cell death. Now, another kind of cell death mechanism is entosis, right? What is entosis? There are some interesting mechanisms in which live cells are engulfed by other live cells, right? Now, these, in most cases, the engulfed cell is usually killed. Right, one such cannibalistic mechanism that is prevalent in many epithelial tumor cells is entosis, in which one cell enters into another cell. Right? So, if, for example, you imagine that the glucose starvation condition is there, and it is a new, this condition is a known inducer of entosis, where the starved cell engulfs another cell to obtain necessary nutrients. Right. So, there's one cell that is starved. It wants the nutrients, so it will engulf a close by cell. Now, this condition in which this engulfed cell is present inside or within this other starved cell is known as entosis, right? E-cadherin is known to assist the cell-to-cell interactions and subsequent engulfment. More recent studies have demonstrated the role of actin and myosin during engulfment. Also, microtubules, microfilaments, and Golgi apparatus are also involved in this form of cell death. The cell thus entered is then eaten up via lysosomal activation. Okay, but it is not always true that this is only what will happen. Interestingly, there are cases where the engulfed cell survives and divides within the host, right? This could also occur. Also, sometimes the engulfed cell can escape and survive. Entosis differs from phagocytosis by the formation of cell-in-cell conformation, right? Over here, not a part of the cell has gone into the existing cell, and it is not always necessary that it will die, right? This cell will be maintained as an individual unit within this other cell, right? Because sometimes it will also, divide within the host. So it is maintaining its own identity within this other cell. So the cell in cell configuration is what is unique in this particular type, that is entosis. Then entosis can be inhibited by ROCK1 or ROCK2 inhibitor and cytochalasin, right? This is an example of or a representation of entosis. So over here in a glucose starvation condition, there would be a glucose starved cell and a viable cell. Now, with the E-cadherin the cells with E-cadherin overexpression will be the cells that will be the viable cells that are engulfed within the glucose starved cells, right? So the progression of engulfment occurs. After the engulfment is complete, what could happen? Either this engulfed cell could escape and it could survive, or it could divide within the engulfed cell, or the death of engulfed cell for nutrient recovery by the glucose-starved cell or entosis will occur, right? So that death of that engulfed cell is actually known as entosis. Then we come to ferroptosis. Now, this is something that most of you might have heard, unlike the previous few that we have talked about. Ferroptosis is actually an iron-dependent cell death initiated and progressed by accumulation of lipid peroxides. It is also present in various degenerative diseases like Parkinson's disease and ischemia or reperfusion injury, right? Ferroptotic cells display smaller mitochondria with ruptured outer membrane. This form of cell death starts with accumulation of iron in cells that result in generation of hydroxyl radicals, and it progresses rapidly when the plasma membrane system XC, or which is a system XE system is a cysteine glutamate antiporter that is crucial for glutathione production and oxidative protection. And this XE system becomes dysfunctional in this ferroptosis. As a result, the cells lose their antioxidant defense mechanisms. Beclin 1, for example, promotes ferroptosis by inhibiting plasma membrane system XC. Specifically during ferroptosis, 5-AMP activated protein kinase AMPK phosphorylates Beclin-1, which translocates to the plasma membrane, promoting pore formation. A combination of the tannic acid and ferric was shown to induce ferroptosis in 41 breast cancer cells. Here, the reduction of ferric led to lipid peroxidation, indirect inhibition of GPX4, and finally induction of the cell death. Elastin, sulfasalazine, sorafenib, and artesunate are also inducers of this type of cell death. Also, ferroptosis can be inhibited by GPX4 and by chemical agents such as ferrostatin and liproxstatin. Why? Now, this is an illustration of ferroptosis, how it will occur, right? Because of basically dysfunction of the system XC. Sorry. And because of eventual increase in the lipid peroxidase and the ROS inside the cell, there will be ferroptotic cell deaths that will occur, right? Then we come to lysosome dependent cell death. We are in the final few slides for today. So this lysosomal dependent cell death, or LDCD, is it is characterized by lysosomal membrane permeabilization, or LMP, and the resultant effusion of lysosome proteases such as cathepsins into the cytoplasm. The elevated levels of ROS, non-lysosomal proteases such as calpains, and lysomotropic drugs such as ceramecin can induce LMP to trigger a cascade of events leading to acidified cytoplasm and eventually to the death of the cell, right? So if the lysosome is basically broken down within the cell, it will be the pH will drop down like anything inside the cell. So probably the cell will also die. The cathepsins released by lysosomes play pivotal roles in inducing the cell death. Cathepsins, especially cathepsin B, has been shown to activate pro-apoptotic proteins such as BID and BAX as well. LDCD plays vital role in conditions such as neurodegeneration, inflammation, host immune response, and cardiovascular disorders. These statins and serpin can block cathepsin and thereby inhibit LDCD. So this is how lysosome-dependent cell death will occur. So if there is permeabilization in the lysosomal membrane, the cathepsins will be released, and due to the low pH, cell death could occur, or it could also go and basically recruit BID and BAX at mitochondrial membrane, which could again lead to permeable mitochondrial outer membrane, which will lead eventually to apoptosis through the intrinsic cell death pathway, right? Now we come to Parthanatos, right? I think this is the final type of cell death that I am discussing today, that is Parthanatos. Now, Parthanatos depends on the activation of PARP1 enzyme, right? So this PARP1 we have seen in other forms of cell death also. We have seen it in apoptosis as well as necrosis. Hyperactivation of PARP1 in response to extreme genomic stress, hypoxia and hypoglycemia leads to ATP depletion and triggering Parthanatos, right? There are specifically after DNA damage, PARP1 activity shoots up nearly 500-fold, generating poly ADP ribose polymers, or PAR. These PAR polymers in the cytoplasm induce the release of AIF. The AIF translocates to the nucleus where it fragments the DNA, leading eventually to the cell death. Parthanatos is independent of caspases, and although it is associated with loss of membrane potential, it does not involve membrane swelling and rupture like that of necrotic cell death. Right? INO1001 and cilostazol are inhibitors of this particular cell death mechanism, that is Parthanatos. Right? This is what it looks like. due to genotoxic stress, excitotoxicity, methyl nitroso guanidine or increased ROS. There could be, DNA damage that could occur, which will accumulate a lot of PARP1. When this is accumulated, it will release PAR polymers, which will go and affect the mitochondria. AIF will also be now recruited, and it will basically be translocated into the nucleus, and this AIF will further lead to chromatin condensation and DNA fragmentation, and all this will lead to cell death, which is known as parthenatosis. So that is it for the various kinds of cell death types, right? So as I said, this is the final practice session pertaining to a particular week's lecture, but it is not the final practice session in totality, right? So we have an extra section, and I have scheduled that for Tuesday, I think from 7 to 9, that is 19th of March, between 7 to 9, we will have a final revision session, recap session on this particular course, that is cell biology. Now, what I can also do in that session, I'll see how I want to plan that session. If the revision is taking longer, maybe I won't be able to do it. But if I will be able to revise in like one and a half, in 1 hour maybe, then what we can do is we can go through the assignments that were actually given for this year also, because till then all these assignment answers will have been released, and then we can discuss the concepts behind those also, so that will also help in your exam preparation. So having said that, that is all from my end for today. I will just stop sharing my screen. Yeah. So if there is a query or a question, you may please ask. Else I will just end the session. Okay, so I have informed you about the extra session that we'll be conducting at the end of this week, right? Not the end of this week, in the middle of this week on Tuesday, that is 19th of March, right? So we'll see how I'm planning that session. Anyway, I hope to see you there as well. And thank you for staying through the entire session. I know that today's discussion particularly was very, very data intensive. I have tried not to do that in the past few sessions. This I just wanted to get across that there are so many different kinds of cell deaths also that we might not know about, because they are not normally, present in our textbooks, because they are so new, right? So you don't have to mug up these various kinds of cell divisions, right? What is required are the major ones, that is autophagy or apoptosis, necrosis, that you should know very nicely what are the characteristics that are associated with them. But I thought it will give you an edge, especially for people who are in their masters and who are maybe planning to stay in academics in the future. It might really help you knowing that there are other kinds of cell divisions as well. Right. So that is it from my end. I'll just stop the recording. And thank you for attending this particular lecture. I'll see you on Tuesday.